Hi, I'm Brian. Along with me is Gabby, Andrew, and Emily. And today we're going to tell you about our part in the MGH purification. We did the SDS page portion of it.
Is okay. So as Bryant mentioned, we did the SDS page, which is basically a gel electrophoresis. The first thing we did was pick the fractions and mix it with the loading dye. We used 10 microliters of the fractions and the loading dye. And then we put them in a heat bath to make sure the stain took, centrifuge them down, so everything mixed back together. And then we loaded them into the vertical gel and started the electrophoresis. And then once the gel was finished, we used the Kamasi blue staining to see the proteins. And then with the samples, we loaded in a ladder as well as pure MGH.
So this is what our gel electrophoresis ended up looking like after the Kumasi blue staining. And then in cells 1 and 2, we have the protein ladder and the authentic MGH, and we put them over there so that they were right next to the fractions that we expected to see the protein in, so that comparison could be done more easily. And then the other fractions to the far right are either ones that we did not expect to see the protein or ones that just looked different after the FPLC and we wanted to take a closer look at.
So these cells, which are 11 through 15 or fractions 6 through 10, we use these because they were the first peak that was seen on the FPLC. And as you can see in the stain, these fractions contain a lot of protein, but no MGH, which is expected because the MGH should still be on the column and should come off later in the other fractions.
And then here is fraction 15 showed against the pure MGH and the ladder. It was the second peak in the FPLC. That's why it was chosen. We wanted to see if any of that protein was any of the MGH like escaping the column, and that's what made the peak, but no MGH was present.
So here's fraction 18 against the ladder and the authentic sample, and it was the third peak that was shown in the FPLC. And like fraction 15, it showed that there was no MGH and also very little of any other proteins as well.
So these were fractions 20 through 25. And we chose these because the green fluorescent protein assay showed MGH in all of these fractions. Fraction 20 or cell 8 can be seen to have little to no MGH present in it at all, but fractions 21 through 25 have higher concentrations of MGH. Fraction 25 or cell 3 can be seen to have the closest to having no other contaminants outside of this MGH. But based on the authentic MGH sample used in cell 2, fractions 22 and 23 look the most like it, and we should look more into things like the yield to figure out which one has the best MGH sample in it.
As for things that we can do to improve the process, the main thing would be running a size exclusion chromatography, just because that could help remove some of the contaminants that are seen in some of the later fractions, and help remove some of the smaller bits of protein that still have the his tag attached to them that aren't the full protein that we want.
So what fractions should we pool? I personally believe that fractions 25 and 24 should be pooled because of their purity. Depending on the amount of yield needed, I would only pool fraction 25 because it was the most pure.
And then I chose to pool fractions 22 and 23 because they look to have the highest amounts of MGH within them.
I said I would pool fractions 22 through 25, and this is because the amounts of MGH in them are pretty high and there's not as many contaminants, but some of them have less MGH that can be seen in them and more contaminants and vice versa. So just trying to figure out like more data on that.
And then I said to pull fractions 25 and 22 because 22 seemed to be the best yield, while 25 was the most pure sample with a little less yield.
All right, and that is SDS page.